Welcome back. We're going to start part 2 with an explanation or description of nomenclature. Classification is very important, and classification is done by genus and species. That's how we report out bacteria in the clinical laboratory. And it's important because we want to make sure that if we have a patient with, for example, E. coli, Escherichia coli, especially think about how important this would be in a contaminated food outbreak, like with the lettuce or spinach or strawberries. We want to know that the same bacteria that's causing a problem in Mississippi, in say Biloxi, Mississippi, is the same bacteria or not that's causing the same type of symptoms and health outbreak in Dallas, Texas. So that's why we want to know the genus and the species. And then if we want further clarification or further connectivity, we would have to send that to, or not necessarily send it anymore because we have molecular capabilities. We can get the genetic sequence and see that it is, in fact, from the same source. So how these are named. The first syllable, sometimes the genera has the same first letter, so the first syllable is used. For example, for the genus Staphylococcus, we abbreviate that staph. And notice how the S is capitalized in the genus and it's written in italics. So to be technically correct, the genus should be capital and italicized, and the species should be lowercase and capitalized. So the abbreviation that we use for Streptococcus is Strept, and for Escherichia coli, we use this first syllabus, Esch, and then, for example, the parasite Entamoeba coli, we just used int. So that's how you'll be seeing this a lot in this semester, and that's what it means. Okay. So slide 9 and onto your study guide. There are 3 categories for bacteria. Correct classification is very important. And the 3 categories that we place bacteria species are family, genus, and species. Taxonomy is the orderly classification and grouping of organisms, and this is based on genotype and phenotype. Genotype is the genetic makeup, and phenotype is the quote-unquote readily observable features of an organism. Examples of the phenotypic characteristics include macroscopic and microscopic characteristics. Macroscopic characteristics include size, shape, color of the colonies produced, or odor of a bacterial colony on a different medium. Staining characteristics, so whether an organism is gram-positive or gram-negative or acid-fast, etc., that's another phenotypic characteristic. Here is a diagram of a cell wall of gram-positive on the left versus gram-negative on the right. The first major step in classifying bacteria is the gram stain, which allows us to differentiate bacteria as gram-positive or gram-negative. So if you look closely at this diagram on the left, gram-positive, the cell wall is made up of a very thick peptidoglycan layer. Let's see, very thick peptidoglycan layer, or a murine layer. 2 components that are unique to the gram-positive cell wall are teichoic acid and lipoteichoic acid. And these compounds are anchored to the peptidoglycan layer. So let's go back and look at these gram-positive versus gram-negative. So here's this very thick peptidoglycan layer, the murine layer. See, there's a murine layer in both gram-positive and gram-negative. But what's important for the gram-negative cell wall is this lipopolysaccharide layer. Okay, so we're going to go on to slide number 12 and talk specifically about the gram-negative cell wall. It is composed of 2 inner and outer layer peptidoglycan layers. The inner peptidoglycan layer is much thinner than that of the gram-positive cell wall. Gram-negative bacteria have an additional outer membrane called the lipopolysaccharide, or LPS layer. The LPS layer is made of proteins, lipids, and lipopolysaccharides that is unique to gram-negative organisms, and they contain 3 regions: the antigenic O specific polysaccharide, a core polysaccharide, and an inner lipid A, also called an endotoxin. And this lipid A endotoxin is what gives gram-negative their special powers. They are responsible for producing fever and shock in patients with a gram-negative bacterial infection. And here's a picture of a gram stain. So we have our gram-positive cocci. These are in clusters all through here. So that would indicate a Staphylococcus genus. So gram-positive cocci in clusters, and that's what we would report to the physician if this came straight from a biological sample from a patient. These are gram-negative rods, these pink rod-shaped bacillus gram-negative rods, or gram-negative bacilli, which would, E. coli would be a bacteria that would stain that way. All right, and we talked about this in Micro 1, the acid-fast stains. Mycobacteria and Nocardia have a cell wall structure kind of like gram-positive organisms, but with an added waxy glycolipid and mycolic acid, which is a fatty acid. More than 60% of the cell wall is lipid and mycolic acid, which makes it very difficult to stain with gram stain. So these bacteria with the fatty cell walls are best stained with an acid-fast stain. The primary stain is carbonyl fuchsin, which is bright pink. The decolorizer used is acid alcohol, and other bacteria are decolorized by acid alcohol, but Mycobacteria and Nocardia are not, which is why they are called acid-fast. The next group is bacteria that lack cell walls, and these examples are Mycoplasma and Ureaplasma. So staining is not going to happen, and it's also important to know when you're working with these microorganisms, because the antibiotics that kill bacterial cell walls are not going to have any effect on these organisms. So it's important to know membrane structure. All right. Some bacteria may have a capsule. This is a major virulence factor. It's the thing that gives bacteria their special powers. This is difficult to gram stain, and this virulence factor is because this capsule helps bacteria evade phagocytosis. And it's important to know which bacteria have a capsule. Remember, we're talking about bacterial structure. We need to know which bacteria have a capsule in case we need to remove it to test it. So this is also called somatic cell wall antigens. We remove capsules in the lab by boiling. We actually have a hot plate, and we have a little flask of water, and we put a sample in a glass tube inside of a glass dish with water and let it boil for 5 minutes, or we can just use a heat block. An example of bacteria with a capsule that must be removed prior to testing is Salmonella typhi. Salmonella typhi has a capsule antigen called the VI. So that's a capital V with a lowercase I antigen that must be removed for us to test the somatic or cell wall O antigen against Antisera, so we can result the type specific of Salmonella typhi. Capsules also make it difficult to gram stain bacteria and can cause a halo appearance around bacteria on a gram stain. Slime layers are similar to capsules, but they are more diffuse layers around the cells. So that's different. All right, this section is 10 minutes, so we are going to pause right there with and end part 2.